Lately, I've had to warn you about all sorts of please don't do that. And I guess now I have to add, don't get bitten 200 times by snakes. But since this one guy's been bitten 200 times by snakes and given himself hundreds more doses of snake venom, researchers are taking advantage of that hazardous life choice to make better antivenoms for the next person who's bitten. But first, let's talk about what venoms are and how antivenoms work. Plants and animals have two broad classes of toxins they make or accumulate specifically to cause harm: offensive and defensive. We sometimes talk about how poisons are defense, venoms are offense, and while that's not always the way those terms are used, it can still be a useful distinction to draw. Plants, for instance, are often more poisonous than venomous. They don't often go on the attack, but plenty of plant species have evolved through selective pressure to accumulate substances that are toxic to animals that might want to eat them. Like, a bunch of plant alkaloids originally evolved as, dude, don't eat me, that were toxic to the animals that want to eat them. Caffeine's a great example. It shouldn't be a surprise that many of our medicines come from plants. Those plants evolved to accumulate chemicals that would specifically act on animal physiology, and lo and behold, we use those chemicals, or related ones, to manipulate our physiology. So the plant has to sacrifice some energy toward these defenses that could otherwise be put toward growing and reproducing, but in exchange, those poisons deter animals long enough to actually, reproduce. Worth. Some of these substances bioaccumulate up the food chain as well. We once covered how the poison dart frog avoids being poisoned by the substances it accumulates, and it originally gets those poisons from eating insects, which in turn either make those toxins themselves or get them from insects or plants that they eat. By contrast, venoms are actively delivered, usually as part of a predatory strategy against prey animals. Venom is usually delivered by a sting or bite or other subcutaneous method, that is, puncturing the skin. And that seems like such a weird detail, but it provides context as to why venoms and poisons are chemically distinct. See, defensive poisons are very rarely proteins or peptides, while venoms often are. A neurotoxic venom, like the box jellyfish or black mambas, is a protein that affects the nervous system, like by disrupting the ion channels that nerves use to signal. A necrotoxin, like some vipers have, causes unprogrammed, messy cell death, causing an inflammatory response with collateral damage that can ultimately result in tissue death. So on and so forth. If these proteins or peptides were used as a defensive tool, most species' stomach acid would just break them down. Breaking down proteins and peptides is a key part of the enzymatic digestion process. It's literally a core part of the stomach's job. So with defensive poisons, it's got to be the kind of molecule that can survive the stomach, because the whole point is to discourage being eaten. But because venoms are delivered through things like bites, they obviously bypass the stomach and get right to the bloodstream to cause problems. All of that is to say, it is not universally true that venoms are proteins or peptides, while poisons are small molecules, like caffeine. But when we understand how venoms and poisons respectively tend to get into their target system, it's not surprising that we see this broad trend. In these venomous species, their DNA codes for the venomous protein or precursor, so you get tons of different variations on venomous proteins through selective pressure. And yes, over time, there's an evolutionary back and forth between venomous species and their prey. I develop resistance, you develop more potent or effective venom that evades my defenses. But that only works at a population level. As an individual, when a bad shape, like a venomous protein, is introduced to our bloodstream, how do we defend against it? With antibodies. The whole point of our adaptive immune system is that rather than have a finite catalog of threats we can respond to, we have a unified adaptive defense system that can respond to novel threats. we have a handful of classes of antibodies that all have this same base structure, this little Y-shaped boy here. At the top, two little arms of the Y is the antigen binding site, where an antigen is this antibody's specific target. So maybe that antigen's the protein code of a virus, maybe it's a peanut protein. It's going to be different for each antibody. And indeed, you'll notice that at the top of those little Y arms are marked the variable region, because while every antibody you make has that same kind of overall shape, the very edges at the top of the Y will be different for each antigen. Don't solve each problem individually. Create a problem-solving system. A lot of times biology is complex and complicated and baroque, over-engineered, but this is such a lovely, elegant solution. Which is not to say it never goes wrong. When your body decides it really needs to make antibodies against your own cells, that's an autoimmune disorder. But defending against a constant barrage of novel threats is a hard problem to solve for, and the antibody system we've evolved is pretty incredible as a solution. So you have these antibodies, and in the little arms of their Y shape are these variable and hypervariable regions that bind to the antigen. Often, but not always, a protein or peptide, and they bind in a lock and key way that's largely unique to that antigen. But the lock and key binding site isn't the whole entire protein that is its target. It's usually about 100-ish amino acids on that target, and proteins can be up to tens of thousands of amino acids long. So, the antibody only binds to part of its target protein. Say we got this light green gross little antigen in our system and we hate it. You can see that our antigen binding site locks in with the very intricate side of that antigen, but not the big round curve part. And yes, this is a cartoon, but it reflects another thing about naturally produced antibodies. They're not just specific to a given protein, they're specific to a given part of a protein. You'd need a different antibody if you wanted to bind to the round part of that light green gross little antigen, right? But why would I want this antibody to bind to one part of a protein versus another? And why do I care for the purposes of being bitten by a venomous snake? Because depending on where the antibody binds on its target, it may or may not stop that protein from doing its little protein thing. Imagine I made an antibody to an antibody. Those exist. We've also just learned about different regions on antibodies, so you already know how different parts of that particular protein work. If the lock and key binding site on my anti-antibody is to the long leg of the Y, that second antibody will absolutely bind, but it doesn't stop that first antibody from doing its job. On the other hand, if the two antibodies were joined at the top of the Y, face to face, I've not just bound that protein, I've completely blocked it from doing its antibody job. I have neutralized that first antibody. A lot of proteins are like that. They'll have different sites that are for different parts of doing its job. Sometimes an antibody will bind to a protein, but depending on which part of the protein it's an antibody to, it may or may not stop the protein from functioning. Antibodies that do block function are called neutralizing antibodies. So, when that venomous snake bites you, it's putting tons of these awful murder proteins into your bloodstream. Your body defends by making antibodies. But even just making an antibody is no guarantee it's going to be a neutralizing antibody, right? So your immune system is on the clock. Make antibody, roll lucky you're generating enough neutralizing antibodies that will actually bind to the venom protein in a way that stops it from being a murder protein, and make enough of those neutralizing antibodies that you can cancel out enough venom proteins that you can survive this whole experience. And when you put it that way, it's not surprising that venomous animals can be so deadly. The numbers game is real unfavorable to the prey. If a bite puts in a zillion protein molecules of venom, each of which are going to damage its target on contact, you need as close to a zillion protein molecules of venom neutralizing antibody as quickly as possible to counter the damage. If a species is frequently preyed upon by a venomous species, that prey might evolve venom resistance, so the venom has less ability to damage the target. And indeed, some venoms get power crept over time as tons of species evolve in ways that make the venom less effective at damaging its target. But hey, bold of you to assume that a venomous species only has one venom for you to defend against. Species of box jelly, for instance, can make up to 170 toxic proteins, which is one of the many reasons why getting stung by, say, an irukandji box jellyfish is basically GG. But understanding how venom works also helps us understand how antivenoms work, and why they seem to be so finicky compared to treatments for other conditions. Antivenom therapy is basically giving you a bunch of neutralizing antibody to a particular venom, giving you a store-bought edge on the venom versus antibody balance. It comes with side effects because your immune system really doesn't like antibodies it didn't make itself, but the therapy is still worth the risk because, deadly venom. So, if you've ever heard that antivenom treatments might not be available in your immediate area, we can understand why. It's because antivenom is an antibody treatment, and antibodies don't stay stable. They expire pretty quickly compared to normal small molecule drugs. We'd have to make a huge amount to keep every hospital constantly supplied with every antivenom antibody relevant to their region. And we can also understand why it's so important to know, say, what kind of snake bit you. Because the wrong antivenom is all allergic reaction downside, no neutralizing the venom upside. Going back to our antigen binding, if I give you a bunch of this therapeutic antibody and you've been bitten by a snake that makes blue square-shaped venom antigen, is that therapeutic antibody going to do anything? No. It's lock and key. It's not going to do anything worthwhile. On the other hand, some antivenoms are what's called polyvalent, meaning they can be used to neutralize multiple venoms or venom from multiple venomous species. Usually it's because the venom molecules have a similar structure, giving the lock and key a close enough fit to multiple similar proteins. In the previous example, I had that square-shaped venom, and that definitely wouldn't have bound. But if we look at the shape of this light blue protein, our anti-light green antibody might still work in a close enough kind of way. This is why you can get some degree of cross-immunization sometimes. Like, if you got the flu vaccine, but they guessed which strain would be popular during flu season and they guessed wrong, you might not have gotten the full protection of a perfectly fit antibody response from the prior immunization, but it's better than if you hadn't made antibodies at all. So to recap, because that was already a lot. 1. Venoms are typically toxic proteins that get into our bloodstream through a bite or a sting. So, unlike defensive poisons, our stomach acid doesn't usually get the chance to break them down. 2. Our adaptive immune system doesn't just have a set playbook, it has a book for inventing new plays on the fly. Things like antibodies. 3. Each antibody has a unique binding site on the little tips of its Y arms, which can typically bind only about 100-ish amino acids of part of a protein. 4. Sometimes when an antibody binds to a protein, it can stop that protein from fulfilling its function. Those are neutralizing antibodies, but not all binding antibodies neutralize a protein. And finally, antivenoms are off-the-shelf antibodies already proven to neutralize a set of venom proteins, but will only work for the right venom. An antibody to the wrong venom does absolutely nothing, and you can get an allergic reaction for no reason. But that all brings us to this week's, oh God, I can't recommend you, as my audience, ever do this, brought to you by snake collector Tim Freed. Freed's been covered in the news because, well, he's been bitten hundreds of times by venomous snakes and has given himself 856 doses of venom. I trust I need not explain further why you should not do this. But also, now that we've learned a little more about how venom and antivenom work, we might understand a little better how he survived this experience. After all, venom isn't like a virus. It can't replicate inside me. If I get 5 virions inside me and they get in my cells, they're going to replicate. Congratulations, now I have 20 virions inside me. By contrast, if I have 5 molecules of venom protein, that's going to stay 5 venom molecules. They're either going to hit their target or my system's going to break them down or neutralize them. Either way, I will never have more than 5 molecules to deal with. So, Freed would give himself very, 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 very little bits of venom, little enough that even if his system totally failed to create neutralizing antibodies, it would still be little enough damage that his body could clear it up. To be clear, that is still risky. Biology is complex. Things go wrong. But, as he considered it, it was a controlled risk. This venom immunization approach is different than what we usually mean when we say something like, the dose makes the poison. That goes back to plant toxins, which have consistent physiological effects that can be helpful at lower or harmful at higher doses. Like, sometimes, make heart rate a little faster is good medicine. And make heart rate go a lot faster is bad medicine. That's dose makes the poison. Makes your cell explode venom is just kind of bad. Like, I'm sure there are some venoms we've managed to harness for pharmaceutical purposes, but mostly they're bad. In this case, the goal of the small dose isn't about getting a physiologically positive effect. It was about limiting the potential damage, so your immune system has a favorable numbers game while working out this whole making antibodies against the venom thing. But anyway, this guy's injecting himself with tiny doses of venom, giving his immune system opportunities to create neutralizing antibodies. He's gotten all those bites from actual venomous snakes, which is, like, this is just personally way beyond my tolerance for YOLO. But it's definitely enough to get him featured in the news. And at long last, we arrive at the researchers who authored this week's article, published in the journal Cell. These authors read about this guy in the news and are like, wow, he should not have done that. But since he did. And then they ask him for samples of his blood in order to isolate his antibodies. And this guy's been exposed to a bunch of different venoms, a zillion different times, so he's got tons of different antivenom antibodies. Some of those antibodies are probably going to bind but not neutralize. Some of those antibodies will neutralize one venom. Some could neutralize multiple, right? Actually, let's illustrate that real quick. Each row here is a short amino acid sequence from a venom protein from a different species of snake. Each column is an amino acid. Remember, each antibody can bind up to 100 amino acids on its little Y-arm, give or take? Blue highlighted sites are sites that have the same amino acid at that point in the sequence across species. For instance, in the third column, we can see every single species here has a C, that's a cysteine, at that position. Looking at this, we can kind of imagine why there's enough similarity that if an antibody targeted this site on a venom protein, it might be able to pick up close enough binding to multiple species. That said, no single antibody is going to neutralize all the venoms because the active site of a protein that makes cells explode is going to be a different shape than the active site of a protein that blocks ion channels in your nerves. But the hope is that maybe over the course of 856 doses of venom from a bunch of different species of snake, this guy has a handful of antibodies that can bind to venom proteins from multiple different species of snake, and enough different proteins like that to be able to protect from a wide range of snake venoms. So, our researchers take the antibodies they've isolated from this guy's blood, and they take a bunch of different venom antigens and basically stick them to a plate. And this part is important. They're not sticking the whole venom protein to the plate. It's an abbreviated version that's pretty much just the toxic part. So if any of his antibodies bind to the part stuck to the plate, those aren't antibodies that bind to some useless part of the protein, because the non-functional part literally isn't even on the plate. Any antibodies to those useless parts will just wash away, they won't stick. And if any one antibody binds to multiple different antigens, there's a good chance that's going to be a broad-spectrum neutralizing antibody. In this cartoon representation of their approach, that's the pink-looking antibody, the one they're going for. Now, in theory, we've gotten from this antibody assay a handful of antibodies like this. They stuck to the toxic part of the proteins we put on the plate, so they're probably neutralizing, and the candidate we chose stuck to more than one protein, so the antibody's broad spectrum. But now it's time to put that theory into practice. Our researchers cook up a bunch of these candidate therapeutic antibodies and give it as a cocktail, along with a little bonus drug that isn't relevant here, and they see if the antibody cocktail can actually protect lab mice against real-life snake venom. Now let's cut to the chase. I wouldn't be telling you this story if it didn't work. It would have just been the story of some schmuck who injected himself with tons of snake venom for no good reason. And the authors of the study say it, so I'm going to say it too. There is no good reason for anybody to do this again. We already have the antibodies. There is no scientific benefit to replicating this experiment. Please don't. In fact, it would have been a wild ethics violation if these scientists had asked Fried to do this. The whole reason this thing could happen is because those life choices were already in the rearview mirror. As far as I can tell from the paper, Fried's self-immunization escapade was a wild run from 2001 to 2018, so we are done now, okay? This cocktail has been tested against all the category 1 and 2 elapid species, so like cobras, coral snakes, black mambas, and so on, not vipers or anything. But it offered complete protection against most snakes. Of course, none of this counters any of the other disadvantages of antibody-based antivenom therapy. Antibodies still expire pretty quickly, and you still have to get that antivenom as soon as possible before the damage is irrecoverable. But holy wow, this is a huge advance in the field of antivenom research. And I hope it's a good explanation of why all this works as well. We talk about antibodies all the time. You can even see them in this old school episode of the Magic School Bus, as Ralphie's body creates anti-school bus antibodies. But understanding how they work unlocks a huge amount of insight into how both our own and therapeutic antibodies actually operate. And in so doing, we can appreciate just how remarkable our adaptive immune system really is. So please don't get bitten by venomous snakes. But for the guy who did, thanks, I guess. I'll see myself out.